Okay, so today is our last day of content before the exam. So wherever we get to today, that is where we will end for material for the exam. We don't have to finish anything we don't get to. We'll see how we do. So next time when I see you on Friday, we'll be doing some review problems, bring any questions you have, and I'll make sure to get up a sort of practice test that I'll put on Brightspace so you can work through what the format will be, kind of what the content distribution will be. And hopefully that'll get us set and ready to go. Okay. So last time when we picked up with talking about the blood, we were focusing in on our red blood cells, right? That's one of our main concepts that we want to know. We saw our red blood cells are called erythrocytes. We know that they're made from stem cells in the bone marrow. And we talked about two proteins that we see in our red blood cells. You know that our red blood cells contain fewer things than normal cells. They don't have a nucleus, they don't have organelles, but they do have two important proteins. They have the hemoglobin and they have that carbonic anhydrase that allow them to carry gases, oxygen and carbon dioxide. We also saw that they have really big surface area for their volume, which gives lots of space for those gases to diffuse into the red blood cells. So today we're going to be talking about where those red blood cells come from, where those erythrocytes are synthesized. So we're diving in onto that first part a little deeper. So last time we mentioned they're going to be made inside the bone, in the bone marrow. But now we're going to pay attention to why. We know that they have a relatively short life cycle. They don't have the DNA or organelles necessary to divide on their own to do mitosis. So we're going to need to be constantly signaling the bone marrow to make more red blood cells. Now that signal actually comes from the kidneys. When we get to the renal section, we'll see that the kidneys, one of their main responsibilities is to filter the blood. So as they're filtering the blood, they can actually tell when there are low oxygen levels. When there are low oxygen levels, we want to get more oxygen to all our tissue. And we've talked about a number of ways that the cardiovascular system adjusts in order to deliver more oxygen to tissue. Another way we can get more oxygen to the tissue is by physically making more red blood cells so that we have more hemoglobin, so that we have more ability to carry oxygen around. If there's low oxygen in the blood, the kidneys notice, and they secrete this chemical called hematopoietic growth factor, which creates another chemical called erythropoietin. So that's the one we actually want to remember for our purposes, is that this ultimate signaling molecule we're trying to get is the erythropoietin. Right? It's folded. So the signal is pretty much coming from the kidneys. Now this tells the bone marrow to start creating these red blood cells. We call this differentiation because there are already those stem cells in the bone marrow. What we want to do is tell those stem cells, instead of becoming some type of white blood cell, instead of becoming a platelet, we want you to turn into an erythrocyte. We want you to become a red blood cell specifically. That process is called differentiation. They're becoming something different, and we're specifying what. So the erythropoietin coming from the kidneys gets into the bloodstream, gets into the bone, tells us turn those stem cells into the erythrocytes, turn them into a red blood cell. When we make the red blood cells, the developing erythrocytes, as they're growing, as they're almost as they're shrinking, right? Because they're losing their nucleus, they're losing their organelles. But as they do this, they're going to be producing hemoglobin, or more of that molecule that's really important for carrying oxygen, although we know it carries a little bit of carbon dioxide as well. And at the same time, they're going to be losing the nuclei and organelles that are in those original stem cells. So they're turning into adult red blood cells. A big part of that is making the hemoglobin. This means that we have specific dietary requirements for making more red blood cells, for making more erythrocytes. And you may have thought about some of these dietary requirements as relates to anemia, which is a condition basically where for various reasons we don't have enough hemoglobin or enough red blood cells. And often this is due to not enough iron. So this is part of why we need to have enough iron in our diet, is to produce those red blood cells and to produce that hemoglobin. Mm-hmm.
Why do you need more of that folic acid?
Folic acid and vitamin B12 are necessary for DNA replication. So that's part of mitosis and the cell division. So as an embryo or a fetus is growing, you have to divide cells a lot. You have to make a lot more cells if you're growing a whole new person. So that's what the folic acid is for.
On the last slide, you said that this stimulating happens from the liver when there's low oxygen. Is that like
kidneys? But yes.
Yeah, kidneys.
Yeah.
Or is that like when you're exercising and you don't have oxygen in your blood, and like how quick is this process? Like this differentiate?
I think this is longer term low oxygen, but I'm not actually sure.
Would be like an example of long-term
Regulation, I suspect so, but I don't know. I don't know that for a fact. But I think given that they have a 120-day life cycle, that would be a kind of crazy thing if you suddenly make a ton of red blood cells when you're exercising. But it does happen when you're like bleeding out, if you're hemorrhaging, it is a response to that. So that might indicate that it's more rapid. so I'm not quite sure. Yeah. I doubt it's on the order of minutes. Okay. Yeah. So we have the iron because we need to make hemoglobin. I'm just going to draw a bad picture of some hemoglobin so we remember what it looks like. So we have the protein part, the polypeptide part. We have those 4 squiggles, whatever they look like, kind of bounce together. Because we have 4 subunits, this actually means that any hemoglobin molecule is going to be able to carry 4 oxygen molecules, and we'll talk about that a bit in the respiratory system. The other thing we're paying attention to, I don't know if it's going to show it well on this screen. But what I'm drawing in now is the heme group. Right, so the heme group is that specific binding point for oxygen. That kind of looks like flowers now. Okay. And inside the center of the heme group is where we have the iron. So there we have a little piece of iron, and that's crucial for binding the oxygen. It's really that iron in the heme group that allows us to bind the oxygen. So that's where that iron is going. Make those heme groups, which have to have an iron in the center. So if we can't make those heme groups with the iron in the center for our hemoglobin, we're going to reduce our ability to carry oxygen. So we do have typical hemoglobin content in the blood, which if you're potentially assessing patients in the future, you may compare to. For our purposes, we don't really care about these specific numbers, but it's somewhere around 15 grams per deciliter-ish. You can see we bracket that for both men and women. So the iron, we need to make the heme groups and the hemoglobin. The folic acid and the vitamin B12 are for cell proliferation, are for cell division. The red blood cells on their own can't do, but those stem cells do divide as we're creating the red blood cells. So that's why we need it for the erythrocyte production. And so as we're making more red blood cells, we're dividing those stem cells into many of the cells in that pathway before they lose the nuclei and the organelles. And we're going to need the folic acid and vitamin B12 for that. So that's why you see that as a dietary requirement of pregnancy and in other parts of life as well, because all your cells need to divide to repair themselves, to repair your body. So these are general requirements for normal function. It's just that they are also requirements for red blood cell production. On the opposite end of the life cycle of an erythrocyte, we're going to filter and destroy them. So after 120 days, they're getting old, they're getting worn out. We're going to filter them out using two organs, the spleen and the liver. The spleen is really our first stop, and the main place we're going to see the real removal. So the spleen, you'll see when you get into the abdomen and lab, has some significant blood flow to it. The lymphoid organ, it's filtering blood. As the blood goes past it, it pulls out red blood cells, and it has a bunch of macrophages in it. So that they're kind of like an immune cell. And those are going to break down our erythrocytes, break down our red blood cells. A little bit later in the pathway, the byproducts of that breakdown are going to get into the liver, which is going to further metabolize the byproducts, so like change those chemicals into some other chemicals. We are going to look at a whole map of how this works. So if you can't visualize it yet, that's okay. So the spleen has those macrophages. The macrophages do phagocytosis, so they basically eat other cells. And in this case, when we're thinking about the death of a red blood cell, the death of an erythrocyte, we're saying they're doing phagocytosis of old, fragile red blood cells, the one that have hit that sort of 120 days. So the main thing that we want to be aware of here is we're not trying to completely throw out those old red blood cells. We do want to keep specific parts. Specifically, we want to keep specific parts of that hemoglobin. Specifically, we want to keep parts of that heme group, especially specifically the iron. And this is so you don't have to be constantly eating tons and tons of iron, right? Once you have iron in your body, you want to try to conserve that as best you can. And you have to eat some dietary iron, but not nearly as much as we would if we were constantly excreting the iron from our hemoglobin. So as the macrophages consume those red blood cells, they're going to break apart the hemoglobin. So the hemoglobin gets catabolized. Metabolism is breaking something down. So we're removing the iron from the heme group in the hemoglobin. And then we do throw out the rest of the heme group. And when we throw out the rest of the heme group, it becomes a new molecule called bilirubin. So if we take the heme group and we remove the iron from a center, so now we have the iron separate, we have the heme separate, this heme part becomes the bilirubin. Now the bilirubin is going to go into the bloodstream. And your liver is going to break it down further. And a lot of it is going to get secreted as bile. So bile is this kind of fluid that you're going to see in your gastrointestinal system. It's actually going to be bright green when you see it in the gallbladder and the liver in lab. So that bile travels through the intestinal tract, so through the GI system. And as it travels, it actually turns brown. So this is why poop is brown, it's that bile, which is one of the reasons that you look at changes in the color of feces sometimes to diagnose specific conditions, because that can tell you about things like the amount of bile, and therefore this could even be telling you things about what's going on in the blood. You can diagnose other things as well, but that's one thing you could be paying attention to. The other place that the bilirubin is going to get excreted is in urine. So it would go through the bloodstream, and then the kidneys would filter it out as well. And there it gives it that yellow color. So it's a slightly different color there. but the iron we're saving. So the heme group was turning into the bilirubin, was turning into bile and getting excreted. But that iron, we're going to save it. We're going to use it to synthesize new hemoglobin in the red blood cells. So we have a problem, right? So the macrophages eating up the red blood cells, doing that phagocytosis, they're in the spleen, right? So at this point, our iron is in the spleen, which is not where we're making erythrocytes. That's not where we're making our red blood cells. Where are we making them? Yeah, exactly, the bone marrow. So we have to get that iron to the bone marrow so that we can make the hemoglobin. So we're going to have proteins that either transfer the iron into the bone marrow or that kind of store that iron for a future point at which time you're going to be making a lot of red blood cells and hemoglobin. So you can store it kind of temporarily as well. So the one that actually moves iron around, transfers it. So it's literally called transferrin. So you can see the word transfer in here, but a sneakier word root here that you may or may not know, this FERR, F-E-R-R. Anytime you see that, that is actually referring to iron. We say something is ferrous, that's referring to iron. And so transferrin is transferring the iron. And it's going to transfer that in a bunch of different places depending on where we're trying to move iron. So one of the places we're going to see it moving it through the bloodstream from the liver, but also from the spleen into the bone marrow. We're also going to see transferrin taking iron out of your food, so out of your GI tract, and also taking that iron ultimately to the bone marrow. So anytime we see iron traveling through the bloodstream, it's going to be bound to this transferrin molecule. So when we see it in our pictures, it's going to be listed as T iron, and what we're saying is that that's iron bound to the transferrin. So we're only going to see that in the blood, but whenever we're moving it around in the blood, it's going to be in this form. We want to store it for later. If we don't need to make a lot of hemoglobin right now, we don't need to make a lot of new red blood cells, we still don't want to throw out that iron. So we can bind it to ferritin. So here we see that fer root again. So the ferritin is this kind of storage molecule for iron. And we see that largely when we talk about it, we talk about it in the liver, but we also see it in the spleen and in the small intestine. So we might see things labeled F iron. And when something's labeled F iron, we mean it's stored bound to the ferritin, which means we're going to see that like in organs, but not really in the bloodstream. Right? So the F iron is kind of staying in the spot. Okay, so now we're going to go through this big picture step by step. And we're going to kind of start at the bone marrow at the birth of an erythrocyte. And as you see, we're going to cycle back all the way to the end and talk about the signaling as well. So the first thing we see happening here, first part of our life cycle, is we have erythropoiesis happening. So that's the word for synthesizing erythrocytes, synthesizing red blood cells from stem cells in response to the chemical erythropoietin. Erythropoiesis, the word for making these erythrocytes. So they're making the hemoglobin, they're losing their organelles, they're losing their nucleus. Then they're going to migrate, so they're going to move out of the bone marrow and into the bloodstream. They're going to circulate around, they're going to be carrying your oxygen, they're going to be doing their job for approximately 120 days. And then when it's time for them to die, the spleen is going to pull them in. So they're going to go past the spleen. The macrophages are going to eat them up, and they're going to pull apart the heme group. So when we see hemoglobin, we see that heme group in yellow. From the globin part, which is more like the polypeptide part, the part that really makes it a protein. The hemoglobin is a protein. So we're pulling apart the heme from the globin. Okay. Next, we have a lot of steps here. So we can follow them, track what's happening to the heme versus what's happening to the globin. So these are kind of two separate steps. So the heme is getting further broken down to pull out the iron, because that's what we really want from this whole process is the iron. And then we put that iron into the bloodstream as T iron to transfer in because we're transferring it. Which leaves us with the other parts of the heme and also the parts of the globin to get rid of. So both the rest of the heme that we talked about, but also happens to be the case that the amino acids from the globin are also involved in this process where we're getting rid of the rest of the hemoglobin molecule. So our bilirubin and our amino acids are both in the bilirubin in our bloodstream. Questions so far about this process? Yeah.
So how does it work? How is it different?
That is a great question. I don't know.
Just curious.
I would suspect that maybe the liver takes over a role because we focus on the spleen here, but we do say that the liver can do some stuff. So I wonder if it can take over this whole thing there.
Like we'll go around 120 days. How does the spleen know like which red blood cells like below to know when?
Again, I feel like I'm always saying I don't know to your wonderful questions in this class, but I suspect that there are signaling molecules that indicate that they're getting kind of fragile. Like I don't think there's like a timer necessarily, but I suspect that probably there's something in the membrane that's getting worn down or something, or maybe they start expressing some type of signaling molecule. That would be my guess.
Most of the blood like filters through like normally, or is this just like something that happens off the side only for the ones that need it? They're like
We don't route blood like separately to the spleen, right? Like it's not like separate red blood cells are going there. They do all eventually go past because of the way the cardiovascular system works, right? So it's kind of just at random, right? Whether a red blood cell is gonna go over to your spleen or whether it's gonna go over, you know, to your heart muscle or your bicep or whatever. But because you're pumping so much blood all around your body all the time, they eventually get there. All right, so now we're tracking the bilirubin, just going to the liver. So the bilirubin is turning into bile. All right. Some of the bile is going to go into the blood, and sort of a sneak peek, that's the part that's going into the urine. The rest of the bile is going to be going down into your GI tract, getting excreted as feces. And we can see here that we also have some F iron in the liver. So this is iron that's been stored in the liver from, you know, a previous cycle or from the diet. And we can see that here we're just saying that that F iron can also be migrated into the bloodstream to give us more T iron if we're trying to make a lot of red blood cells. All right, so here we see they've now drawn on that whole pathway of getting rid of that bile over here. All right, so that's just excretion of the bile in the feces. The other thing we've added on at this point is we see that we're getting some dietary iron as well. So we're ingesting some iron. And it's getting absorbed in the small intestine. And we're gonna move it around in the blood again. So this is another way we get T iron into the bloodstream. And all the T iron ultimately is going into the bone marrow, whether it's from the breakdown of a specific old erythrocyte, whether it's something that's been stored in the liver as F iron, or whether it's something you ate. Like ultimately, the goal of that T iron is just to get it to the bone marrow. So now, to get that file over here, so what we've done is track like this biomolecule that we had coming from the liver into the bloodstream, and it's circulated all the way around, and now it's coming into your kidney. So that's not like a new biomolecule. This is just showing us the kind of other side of what's happening to the bile. And that bile is getting excreted in the urine. Finally, the kidneys are also, as they're getting rid of this bile, they're checking what our oxygen content of the blood is, and they are releasing erythropoietin, telling us it's time to do erythropoiesis, time to make new red blood cells. So that EPO is erythropoietin, which I can maybe spell. Did I get that right? How'd I do? I think that's right. All right. So questions about this overall process?
So the erythropoietin, that just is like a signal for them to like make more.
Yep.
Okay. Is that from?
I think it would count as a hormone. Yeah. Oh, right, with the iron in the bone. Like physically how? Yeah. Ultimately, to get into a cell, there's probably going to be a receptor to pull it in. Bone itself is vascularized, so like there is blood flow like all the time into your bone because it's a living tissue. So we don't really need like special routes to get blood there. So I suspect it's coming with the usual bloodstream for the bone. If you look at a bone really closely, like really closely, there are like tiny holes in them called nutrient foramina, which are for the little blood vessels that go into the center. So that's our life cycle. Okay. Here we have an explanation of some different types of anemia. So we have different types of anemia that are going to sort of damage our ability to carry oxygen because we've damaged in some way our red blood cells and our hemoglobin. So the most common type that we talk about is dietary anemia. So it's when you're just not taking in enough iron. So we can keep some of the iron from our old red blood cells, but we do need to be replenishing our iron stores regardless. It's not a perfectly efficient process. So if you have dietary anemia, you're not getting enough iron usually. A sort of subcategory of this is something called pernicious anemia. It's kind of a type of dietary anemia, but it's specifically if you're not getting enough of the vitamin B12. So if you're not getting enough of this vitamin B12, which should be included in your diet, this means that you may not be able to divide cells, which means you may not be able to produce red blood cells, which is not great. Also leads to anemia, reducing that sort of carrying capacity. I'm going to jump down to the end of the list next, so the dietary anemia. The other sort of major example we talk about is hemorrhagic anemia. So if you are bleeding out a lot of blood, right, you have some kind of cut, damaged an artery, you have low blood volume. This means you're also reducing the number of blood cells because they have physically escaped your body, right? So we don't have the red blood cells because they're on the floor or something. So this means we also have a reduced ability to carry oxygen in the blood just because we've reduced that number of red blood cells, and therefore the amount of hemoglobin that was in those red blood cells has also been reduced. So that's a second type of anemia we could have. Now we'll do the, I would say, rarer types of anemia. So in hemolytic anemia, lysis, right, means breaking something down. So there are diseases that specifically attack red blood cells. One of those is malaria. So that would actually be sort of lysing, sort of breaking apart those red blood cells pathologically. And within this category of hemolytic anemia, we also do consider sickle cell anemia as kind of a subset. Sickle cell anemia isn't really an attack on your red blood cells so much as it is a misshaped red blood cell. So in sickle cell anemia, this is a picture, red blood cells, instead of being their sort of donut shape, that biconcave disc, they're like a little crescent moon. And this shape comes along with changes in the shape of hemoglobin, which reduces its ability to carry oxygen. It kind of crystallizes. And also these crescent-shaped red blood cells, because of that shape, sometimes get kind of like stuck in places like your capillaries. So this reduces the ability potentially to carry oxygen because of this shape. However, it does also have beneficial characteristics, which is that like malaria isn't good at attacking sickle cell blood cells. It's really looking for those normal round red blood cells. So it does kind of protect you from diseases as well. So kind of a trade-off depending how common malaria is where you are. Okay. Next, we have aplastic anemia, which is a bone marrow deficit or a bone marrow defect. Anybody want to tell me why that would lead to anemia? Yeah, right. The bone marrow is what's taking or what's making the red blood cells. If you have a bone marrow defect of a specific kind, it might damage your ability to make new ones. Okay. And in renal anemia, what do you think might be going on with renal anemia? Which renal always refers to your kidney. Where did we see the kidneys in this process? Yeah, kind of like in the signaling process with the EPO, the erythropoietin, right? So if we damage the kidneys, we might damage that sort of filtering of blood process and also damage potentially our ability to signal to the bone marrow to make more red blood cells. So basically, we can get anemia if we attack kind of any part of this process, right? Like if anything goes wrong here, we may end up with fewer red blood cells or fewer hemoglobin molecules. In certain types of anemia, you actually see like giant red blood cells, but not enough of them. Okay, so we'll do a couple practice questions. We'll have lots of practice questions on Friday. Thinking about the blood cells that we're making, response to hematopoietic growth factor, and we're specifically thinking about erythrocyte production, which should let us know that we're looking for that signaling molecule that comes from the kidneys, and its name is erythropoietin. So the spleen is breaking down red blood cells, right? red blood cells. We'll see that blood vessels, right? We can think of them as either carrying blood, right? They're part of our cardiovascular system. And we'll also see that we're going to talk about the blood vessels and their role in clotting. And the liver is more involved in storage or breakdown. Right? So storage, when we think about the iron, or breakdown and excretion when we're thinking about like bile. Another type of question we could have is just overall kind of top-down view of where the red blood cells are being born and where they're dying. Someone let me know where they're being born. Where are we doing the synthesis, the erythropoiesis? Yep, in the bone. Okay, so that leaves us with either C or D because we're in the bone marrow. And where are they mostly dying? Yeah, dying. I will, after class, look up what happens if you get your spleen in, because I do not know. So we're skipping the part on leukocytes because hopefully he talked about white blood cells in his lab last semester. But we will see occasionally a couple parts of white blood cells in our future units. We've talked a little bit about macrophages here in the spleen as being important. We'll talk a little bit about immune function in the lungs in our next unit. But pretty basically, leukocytes, leuko means like white or pale, looks Greek to me. They're defending you against pathogens of various types, and they're also basically on cleanup duty, breaking down old cells. Our final topic we're thinking about is hemostasis. You want to be careful when you see this word because it's not homeostasis. Hemostasis. Whenever we see hemo, hemo means blood. Like we saw hemolytic anemia on one of those previous slides because we were breaking down blood cells. So hemostasis is going to be basically keeping your blood inside your body, keeping the right amount of blood. So what we're really talking about for our purposes right here is what happens if you get a cut, a laceration, you start losing blood. How do we stop that process? How do we keep our blood inside? How do we keep our blood volume? So we're going to be talking about clotting. So for this process, we're basically going to be having a series of steps, and then we're going to dive in deeper for each of the steps. So this is our first kind of slide where we could consider 3 steps. So the first thing we're going to see happen is we're going to see a vascular spasm. We're going to see that the blood vessels physically respond to injury. Then next, we're going to see the role of platelets. So the second big picture thing that's going to happen is we're going to have platelets migrate to the cut and form a plug. So they're going to be like the first little stopgap in that cut. And then the third thing that's going to happen is we're going to form a bigger, tougher, stronger blood clot. So blood clot is not made up of platelets, it's made up of basically a protein, and that's what is physically able to form something like a scab to actually truly prevent the escape of blood. So these are not different mechanisms, they're all part of a process. Let's start with the vascular spasm part. So whenever you see this word spasm, a spasm really is like a contraction. So if you have a spasm in a muscle, right, like a muscle spasm, that is like a tensed up kind of almost like a cramp, like a tensed up muscle. So vascular spasm in the vasculature, the blood vessels, is sort of constriction, a sudden constriction, a spasm, clenching of the blood vessel that's been injured. And this is an intrinsic vascular response, meaning it's automatic and it's happening right at the level of that injury. Right. What's happening here, right, is as we cut the wall of the blood vessel immediately responds by contracting. There's also some sympathetic innervation kind of amplifying this effect. We'll see some of the same sort of neurotransmitters from the sympathetic nervous system aid in this constriction of the blood vessel. So thinking back to the other topics we've talked about, why would this vascular spasm minimize blood loss? It reduces flow. How does it reduce the flow? Which is affecting, like, what physical property are we interested here? So, yeah. So it is affecting the flow, but the reason it's affecting the flow is that we have that equation, which is our flow, is our pressure divided by our resistance. So that spasm, that vasoconstriction, acts on the resistance, because as radius goes down, resistance goes up. So everything all of you responded was correct, but it's really like this is the basic reason this is happening. Those are all kind of downstream effects. So really, it's just like vasoconstriction where we saw it elsewhere, right? We talked about it in the arterioles. When we constrict, it raises resistance. We did talk about the pressure more when we talked about venomotor tone, which was also kind of a constriction. But that is because all of these factors are kind of related to each other. So this is what the spasm is doing. We're reducing blood flow by increasing the resistance because a constricted vessel is smaller and has a smaller radius. The other thing that is happening, and the next thing that's happening as we do this vascular spasm, is that the endothelial layer of the blood vessel is becoming sticky. So the endothelial layer is going to be a layer of cells inside the blood vessel. And we'll take a look at the picture of a blood vessel wall in a second. So that's going to be part of what's creating this next part, the platelet plug formation. The platelet plug formation is going to have steps within it as well. And just be aware of that. So our platelets are also known as thrombocytes. When you ever see this part, a thrombus is a blood clot. Thrombus is a blood clot. So there are cells that start this process of making a thrombus, of making a blood clot. The first part of this process is that we're forming this plug around the place where we damage the vessel. So we're going to see that there are different molecules that are going to be released by the cells in response to the vessel damage, which are going to make that endothelial layer sticky. It's going to cause platelets to adhere to it, to stick to it. And then they're going to aggregate, they're going to lump together to form a blood clot. And specifics of that are also on future slides. But this plug, though, plugging up the gap, that laceration, that site of vessel damage, which further decreases blood loss. So we've reduced the flow to the blood vessel with the vascular spasm. And now we're starting to cover up the hole, which is going to further reduce blood loss by actually preventing blood from getting out a little bit. The platelets aren't super effective at creating this decrease in blood loss. So we are going to form a stronger blood clot after the plug is formed. But this is the first step. Couple facts about platelets. Platelets are basically cell fragments. So like the red blood cells, they're going to have no nucleus, but they do have organelles and granules we've seen in lab, but they're going to be able to secrete stuff, basically. That's the point of that. They're little cell fragments. So here we have our sort of subset of the platelet plug formation, the platelet adhesion. So when the blood vessel gets damaged, the layer under the endothelium, and I'm going to be careful about my direction words here because it kind of depends whether you're thinking about it from the perspective of a blood cell or from an observer looking out. So the subendothelium is going to be farther away from the blood, which we'll see in a picture. But when we damage the blood vessel, this exposes this layer, this subendothelial layer. And this causes that layer to release something called von Willebrand factor, which is abbreviated VWF. Von Willebrand factor is going to bind to collagen fibers, which are also in those blood vessel walls. So they're also part of these layers of the walls of the blood vessel. And that's the stickiness, basically. So the von Willebrand factor is making the collagen and the endothelial layer sticky, which is causing platelets to stick to it. Adhesion, like an adhesive, like tape that's sticky, sticks stuff together. Adhesion is the platelets sticking to the blood vessel walls. And the platelets at the same time are going to be secreting different messengers, different chemicals, different molecules that are going to cause basically a positive feedback loop of more and more and more platelets sticking in. So we had adhesion. Pay attention here because we have another A word. So the adhesion was the sticking to the walls of the blood vessels. Now aggregation is going to be the platelets sticking to other platelets. And this is basically what creates the platelet plug. The plug is a bunch of aggregated platelets. So we have a couple molecules that are doing this. So we had the von Willebrand factor released by the vessel walls. The platelets themselves are releasing a couple of molecules that increase the stickiness and tell more and more platelets to stick in. So specifically ADP. And this other molecule called thromboxane A is also involved in this feedback loop. So these two are what's creating the platelets to stick to each other. And then we see a couple of other molecules involved in this process as well. So at the same time, platelets release serotonin and epinephrine. Epinephrine we've seen before, and we know causes vasoconstriction as it binds to alpha receptors. Serotonin we haven't seen as relates to the cardiovascular system. And the role of this serotonin from the platelets is not to make you super happy. Serotonin in the cardiovascular system causes vasoconstriction. Then I'll also be releasing chemicals that are going to facilitate forming of the blood clot afterwards. It's going to be the kind of next step, but we will not be getting to the next step. Right. So we're going to leave off. I'll cover through the formation of the platelet plug for the exam, and we'll leave the formation of the blood clot for next time, which I think is good because that has a kind of scary looking map, which is worth more than a couple minutes. I mean, have you looked at it? Have you looked ahead? This scary map? This? We don't want that on our exam, honestly. That can be for later.